Alright, so hey, welcome to our second video. Um, so it's about the scientific method, so we'll jump right in and see how far we can get. Alright, so scientific method is kind of how all of science goes. However, it's not written in stone. And most people think in order to do science, you have to do an experiment. But you don't. You don't have to do controlled experiments to do science. You can do observations and do science. You can analyze things and do science. Okay, so scientific method is a general outline, but it's not an end-all be-all, and I'll probably mention that a lot. So to start with science, we've got to start with a question. Okay, all science has a problem or a question, and these questions usually come from observations about the natural world. Okay, so you see something, you don't understand that something, and then you want to figure out that something. Now you have a problem. So you form a hypothesis. You come up with some sort of solution, explanation, way to show how that's working. Okay? Hopefully it's right, but if it's not, that's cool. What you're going to do next is test that with a controlled experiment. So somehow go about testing it. Either observe when what you think is going to happen and see if it does, or set up a way to make it happen and see if it happens. If it works, awesome, you solved the problem. If it doesn't work, awesome, you figured out something that doesn't work, and we can move on from there. So, in order for a hypothesis to be useful, all it's got to be is testable. And it doesn't have to be right. There's a lot of hypotheses that are not right, but they've got to be testable. Without being able to test them, there's no way to know if they're right or wrong. Okay? And again, we test hypothesis with controlled experiments. So to figure out whether or not your hypothesis is actually working or not, you've got to have everything the same except for what you are testing. So we call that controlling or having a control. Okay? All the variables are kept the same, except for one. You got to change one variable. So you're testing plant height with growth with sunlight, change the sunlight. But you got to keep everything else the same. Amount of water, the temperature, and the nutrients in the soil. All of those can affect plant growth. So if you're just testing light, you got to keep everything the same. Okay? A control group is the group where you don't even change the manipulated variable or the thing you're testing. It's just the same as normal. Okay? That one thing you're testing, the one thing you're wondering if it changes, is the manipulated variable. Or some people, if you look online or look at other resources, will call that the independent variable. That's the thing you change. That's what you're testing. So in, say, plant growth and light, it would be the light that's manipulated. I'm either going to have light or not have light. And the one that responds is the responding variable. Another way to remember that is the thing you measure. So in the experiment I was briefly talking about, plant height versus sunlight, the thing I'd measure would be the plant height. That responds to the amount of light taken in. Okay? Some people call that the dependent variable. All right. Some beginnings of the scientific method. Some people that kind of started these controlled experiments to prove or disprove things. One of them was Redi. Okay? In the time that Redi lived, one of the common misconceptions was the idea of spontaneous generation. Life came from non-life. Okay? So if I want some maggots, I put some meat on my table. After a few days, maggots form. Okay? If I want some mice, I put some old clothes in a bucket in the corner, and eventually I get mice. Okay? Redi, on the other hand, thought, hey, I don't think that's how this works. I think the maggots come from the flies that get on the meat. And so he wanted to test that. So he thought up an experiment. He had 2 jars. One jar had meat in it, one jar had meat in it, and a cover on it. Now, he probably ran this with a bunch of different jars at a bunch of different times. Replication is a good thing to do. But to simplify it, we have a jar with meat and no lid, a jar with meat and lid. Okay? Now we can look at this and figure out the controlled, manipulated, and responding variables. Okay? The manipulated variable, the thing he changed, okay, or the independent variable if you have the other terminology, is the lid. One of them had a lid, one of them didn't have a lid. Okay? The controlled variables would be everything else that he had to keep the same. So the same type of meat, same temperature, same area, have them next to each other on a desk, same access to outside air, all of those other things he had to keep exactly the same. And then the responding variable, what changed based on the lid or not lid, was the appearance of the maggots. So were there maggots or were there not maggots? That is the responding variable, okay, or dependent variable. He found that in the jars he covered, there were no maggots. So he very much supported his hypothesis that the maggots came from flies, because in the covered jars, no flies got there. No flies, no maggots. Flies, maggots. Great! Well, this wasn't the end of the story. Okay? The next thing you have to do with an experiment is you got to analyze it. Okay? So he recorded his results. He did it by hand. Nowadays we do it by computer with calculators, and he drew conclusions. His conclusion was to support the hypothesis. He hypothesized that flies came from maggots, covered jars, no flies, no maggots. Okay? The beauty of science is that if the world really does follow patterns, which we assume it does, then we can draw broader conclusions. So if maggots have to come from flies, then mice probably don't just come from my dirty clothes in a bucket in the corner. They come from other mice. Okay? But people had problems drawing out his conclusions from maggots to other forms of life, and especially microbes, which we'll get to in a bit. Okay? After you analyze the experiment, you have to publish it. Now, nature behaves consistently. That's another one of the assumptions of science. And therefore, no matter where they did Reddy's experiment in the world, it should come out the same. If you covered a jar and flies couldn't get to it, maggots wouldn't form. If you had meat in a jar and flies could get to it, maggots would form. So he had to publish and tell other people about his experiments. Today we do this in the form of scientific journals, where other scientists read your work, critique your work, and if it meets the standards, they publish it. If the other scientists can't find anything wrong with it, they can find things wrong with it. They might send it back for you to redo, to tweak some things, fix some things, and then publish it. Okay? This also allows other scientists to test it. Once you publish it, if somebody doesn't quite believe you, they can do it again. Okay? Or if they think some other result might happen if you change one of these small variables, then they can test that. All right, so people didn't like extrapolating Reddy's experiments, and especially when in steps Leeuwenhoek and finds microbes. Okay, he didn't necessarily invent the microscope, but he invented really good ones, and he could see microbes, little animal, single-celled organisms in pond water. And so various people went about testing Reddy's analysis with microbes. Their names were Sponzini and Pasteur. Okay, they both tested Reddy, but in two different ways. Okay, so Spallanzani simply took flasks, boiled the water in the broth in the flask, so there's some nutrients in it, and then sealed them. Okay, he figured out if he boiled the broth, it killed everything in it, and then if he sealed it, nothing else would get in. Okay, people thought at this point that if you had broth sitting on the table, spontaneously microbes would appear in it and spoil the broth. He thought the microbes were coming in from the air, so if he covered it, they couldn't get to it. Well, it was great. Supported his hypothesis that the microbes came from the air. However, people said, yeah, but you covered the container, so there's no air in it. They didn't know about oxygen, but they knew things needed to breathe air to live, so they said they can't breathe air, obviously nothing lived. So while Spallanzani did support his hypothesis, there were some flaws in his experiment that people hit on. People really grasped at those flaws. So in steps Pasteur. Pasteur wanted a way so that air could get into the container, but the microbes couldn't. So he created what was called a swan neck flask. And actually created a lot of different versions of this, but basically it ran the same way as Spallanzani's experiment. Put broth in, heat broth, kill everything in it. And then, while air can get into it, oh no, I have half a minute left. While air could get in, microbes couldn't. So when he heated these, the broth didn't spoil until he snapped off the lid. Once he snapped off the lid, microbes could get in and it spoiled. Okay? We will pick up on when experiments aren't possible in the next lecture. Thank you very much. and you're welcome for this being 10 minutes.